Hello everyone and welcome back to chapter 19. This is Genetic Technology 2, Recombinant DNA Technology, or it's also called gene cloning. And its function, like the name gene cloning, is to make many copies of one gene. So that's what gene cloning is referring to. But this technology can also be used to make many copies of a single protein. So one gene or one protein. PCR only can make many copies of one DNA fragment, okay, so it's not protein, but here you can do both. Now we'll see the general steps first, and then we'll go into detail for each step. So here are the general steps of recombinant DNA technology. Number one, you have to identify or isolate the mRNA or DNA you want to amplify from the organism. In this case, it's insulin gene. Insulin gene, it can be transcribed and translated into the insulin protein, which is a hormone used in glucose concentration homeostasis, right? You have learned this in your previous chapter. So you just take that gene, isolate it from the organism. You get a plasmid as well from bacteria, or nowadays we just order it online from the manufacturer. Okay, and what we're going to do is want to cut it. So we cut at specific locations, and then the next part is we stick it together. So basically we're putting the gene into the plasmid by cut and paste method. This process is called recombination. So what results is a recombinant plasmid. And this modified plasmid or recombinant plasmid is then placed into a recombinant bacteria. So, it's placed into a bacteria to become a recombinant bacteria. However, this process of insertion into bacteria is not always successful, so we need to identify which ones are modified and which ones are not, and separate those out first. And then we take the modified bacteria, and then we can put it in a large fermenter and grow them, allow them to divide, and use them for large-scale production. As the bacteria divides by binary fission, okay, if we are using E. coli, which is a common bacteria used in recombinant DNA technology, this would double every half an hour, okay, so it divides once every half an hour. That's really quickly. So as it divides, it would copy the plasmids as well. So you can see here a lot of copies of the plasmids have been formed. So as a result, you have many copies of the insulin gene. But not only that, the bacteria also has transcriptional and translational machinery. So they can use the insulin gene you have inserted to produce the insulin protein. And therefore, as a result of this method, recombinant DNA technology, you get many copies of the gene and many copies of the protein. Very interesting, right? So that's how you produce insulin in bacteria. So this is just the general overview though. There is a lot of detail that goes into each and every of this step in order to make it successful. So let's use again the production of human insulin, right? Let's use the idea of that. Let's continue this example and move through step by step and look at the details. Now for human insulin in particular, we usually use mRNA. We obtain mRNA from beta cells of islets of Langerhans of the pancreas, because obviously beta cells are the one that expresses human insulin. So the gene for insulin would be transcribed into mRNA and then translated into the protein, which is insulin, right? So you would expect misspelling there. You would expect beta cells to have this mRNA for human insulin. So you pick a few beta cells, you isolate the mRNA using some technique you don't need to know, and we are going to try to convert it to DNA. And the way we do that is to use two enzymes. Number one is reverse transcriptase. So we use reverse transcriptase to make what we call complementary DNA. And complementary DNA is complementary to mRNA. So mRNA is used as a template to form cDNA. So mRNA would have all the traits you think of as mRNA, right? It has uracil, it doesn't have thymine, it is single-stranded. cDNA does not have uracil, it has thymine, and is complementary, but it's still single-stranded at the moment. DNA has to be double-stranded in this procedure. So we're going to use DNA polymerase, which is another enzyme. You have heard of this enzyme. DNA polymerase is used to make double-stranded cDNA. And now it's complete, right? You have converted mRNA into double-stranded cDNA. However, sometimes you only obtain very little of this, so you only get very few double-stranded cDNA, and you might need more to do your procedure. So how do you do that? You have to amplify it. You can amplify it using the polymerase chain reaction. We learned this in a previous video, by the way. Okay, you might be wondering though, like, why use mRNA of the beta cells? Why not just take the gene? Why not just take the DNA from the beta cells? Or from any cell? Why do we need to take the mRNA? Now the reason is genes are quite complicated, and they actually have many introns, which are non-coding regions. So like extra sequences within the insulin gene that might, interfere with the processes. So we take the mRNAs to make sure we get the coding region only, because only the coding region will be transcribed, and therefore the result is a shorter DNA without all the unnecessary things. That's why we take the mRNA. Anyways, that's a side point. The point is, now we have mRNA and we converted it to double-stranded cDNA. Nice. What's the next step? The next step is to cut, and the following step is to paste. So how exactly do we cut DNA? We cut the gene and the plasmid DNA using restriction enzymes. Restriction enzymes cut at restriction sites. So restriction sites are present at both sides of the gene, of the target gene, and also plasmid, which are obtained from bacteria, also has one restriction site, and this is using the same restriction enzyme. So again, two sites, both sides of the gene, two restriction sites, one here and one here. In this case, they're using E. coli, EcoR1. Sorry, I like to call it E. coli. It sounds like E. coli. It looks like E. coli. Never mind. Anyway, EcoR1, it is a type of restriction enzyme, and cut at two sites of the target gene. Okay, not cutting the gene itself, but just at the two sites, and then also cutting at one site on the plasmid. This creates complementary sticky ends if you use the same enzyme. Why is that, and what am I talking about exactly? Okay, so let's delve into more details about the restriction enzyme, shall we? So restriction enzymes, or restriction endonucleases, fancy name, same thing, right? They are from bacteria. They recognize, bind, and cut DNA at a specific sequence, and this specific sequence is what we know. It's called a restriction site. So again, restriction enzymes cut at restriction sites. Now, what the restriction enzymes do is that it hydrolyzes or cleaves the phosphodiester bond between the nucleotides. Don't forget, the phosphodiester bond is the one that is holding the phosphate backbone of the DNA together. Alright, so the restriction enzyme does that, and there's actually many types of restriction enzymes, okay? Different kind of restriction enzymes will cut at a different specific sequence. So different restriction enzyme, different restriction sites. There are many types. You already saw one just now, EcoR1. This is Hind tree PST1 and EcoR5. I think that's 5. Okay, my Greek number is not that great. But anyways, yeah, there are many types, and they all cut at different restriction sites. Now, restriction sites are mostly palindromic. And what that means is that you can read it same forward and backward. So yeah, you can see this particular restriction site from Hind tree, right? AAGCTT, if you read it this right, from left to right. If you read it from right to left at the bottom strand, you can see that it's the same, AAGCTT. This is the same for PST1 restriction site. If you read it from left to right, CTGCAG, and then if you read the other strand from right to left, it's again CTGCAG. How cool is that? Okay, in English, there are many words that are palindromes as well. Let me Google search a few and give you a few examples. Hang on a sec. Let's Google search. By the way, every time I open a new tab, they give me a new cat. It's an extension. Okay, don't judge me. Little things that spark joy. Let's look for palindrome examples. Some examples of palindromic words are like redivider, reviver, rescar, madam, refer, and it's like refer, it spells R-E-F-E-R, forward, and R-E-F-E-R, backward. So that's what palindrome means, even in English. In this case, we're talking about restriction sites. But it's the same idea. Forward and backwards, it's spelled the same. Okay, on the reverse strand, anyway, for restriction sites. Now, restriction enzymes can produce sticky ends or blunt ends at these restriction sites. What are sticky ends? So at sticky ends, this is actually a result of a staggered cut. And at the sticky ends, there are a few unpaired nucleotides as a result of that staggered cut. So you can see example right here for hind tree. You can see that the staggered cut is like a zigzag cut, right? It's not like a straight clean cut, and what it results in is these sticky ends that are unpaired. So TCGA here are unpaired nucleotides, and they can bind, they can complementary base pair with AGCT. You can see here AGCT again is unpaired, and it can feel free to complementary base pair with TCGA. Okay, so it can stick back together or stick with something else which has those sequences. You can see the same thing here for PST1, right? This is a staggered cut again, a zigzag cut again, and you can see sticky ends produced here. Again, TGCA is unpaired and is free to complementary base pair. Is able to form hydrogen bonds easily with the complementary bases. So that's sticky ends. What we want is sticky ends. This is what we desire. But not all restriction enzymes do this. Some of them produce blunt ends instead. And at blunt ends, there are no paired nucleotides. So it's pretty much a clean cut like EcoR5 here. It's a clean cut, and therefore there's no unpaired nucleotides, and these fragments cannot stick back together. They are not complementary base. There are no unpaired nucleotides that are ready to complementary base pair, so they won't be able to be stuck back together. This will be very important later on. Okay, production of sticky ends will become very important later on to actually insert the target gene into the plasmid. Now, just a refresher, we are using the restriction enzyme to cut the gene at both sides of it, right? Not cut the gene itself, but both sides of the gene, and at one point in the plasmid, using the same restriction enzyme. If we use the same restriction enzyme, then we will have complementary sticky ends that are produced, and therefore this gene can then stick, so-called stick, to the sticky end in the plasmid. We'll see a diagram for it later. But there's one more question here that we need to ask and answer. And the question is, why use plasmids from bacteria? Why can't we just use linear DNA? Why can't we use like normal DNA from human beings? Can't we just like put the entire insulin gene in? And the answer is no, we can't. We have to use plasmids, and for very good reason. Just a reminder, plasmids are small, circular, double-stranded DNA, and they're usually found in bacteria, but now they can be artificially made in the lab, and you can order them online, and then the manufacturer will just ship it to you. It's incredibly convenient. Anyways, you use plasmids because of these benefits. It's frequently used because it's small, so it can be easily inserted into cells. It's circular, so it's more stable. It is not damaged by host cell enzymes. Plasmids are a great vector to deliver desired genes to the bacteria because it's easy to extract and easily taken up by bacteria. What is a vector, you say? You have heard vector in the context of malaria when the mosquito was used to transfer the Plasmodium parasite from like human to human. That's a vector. But in this case, a vector is not used in a disease context, but using a vector is talking about delivering the gene. So the vector is actually delivering the gene we want into the bacteria. So that's why we call plasmids a vector. We will see the word vector again in the next lesson, next video. But anyways, plasmid is a great vector to deliver the desired genes we want into bacteria. Why? Easy to extract, easy to be taken up, has a high copy number. So you don't need to have only one plasmid per bacteria. It's not limited. They can have many copies in one bacterial host cell, and it can replicate independently within bacteria. So even if you insert one plasmid into the bacteria, it can replicate inside the bacteria to have many copies of it. And when the plasmid containing our gene is replicated, it's going to replicate our genes. So it's able to clone our genes, which is what we want the technology to do, right? So that is great. Great, thank you, plasmids. Now, last but not least, actually this is the most important point, it has specific DNA sequence we need for not only insertion into the bacteria, but expression of the gene we want. So we want not just the gene to be cloned, we want the protein product. So we need specific DNA sequences, okay? So what are these specific DNA sequences? I'm glad you asked. So here is a table showing you all the specific DNA sequences. Let's go through it one by one. A lot of words, I know. Don't be scared. So number one is the origin of replication. You need this. If you want the plasmids to replicate independent within bacteria, you need the origin of replication, which is a specific DNA sequence for the DNA polymerase to bind and replication to be initiated. If you don't have this, the DNA polymerase won't bind and the plasmid won't be replicated. You don't want that. You want it to be replicated. Number two, restriction sites. Restriction sites is obviously the site where the restriction enzyme cuts. And where it's cut, you can insert the gene of interest into it. There can be multiple restriction sites in one plasmid to be cut by different restriction sites, different enzymes. Okay, so different restriction sites, different enzymes. And this is good because this gives us choice. This means we have multiple cloning sites, which means multiple sites we can insert our gene into. And this is just great for choice. Number three, promoter. We need a promoter. Again, we don't just want replication, or we don't just want to insert the gene into the bacteria for fun. We want the gene product. We want the protein to be formed, and to have that, you need a promoter. And promoter, same function as you have learned before. This initiates transcription because it allows the binding of RNA polymerase and transcription factors to the promoter. And through this process, this target gene that is downstream of this promoter will be expressed, transcribed into mRNA. Now, promoter also ensures the correct strand is used as a template. What does this mean? Don't forget that DNA is double-stranded. Plasmid DNA is also double-stranded. So which strand is the coding sequence on? The promoter will tell the bacteria this. Not only that, promoter, it can initiate transcription, but it also can determine different levels of transcription and where and when it is expressed. Okay, so it can ensure like a very high level of transcription, or maybe very low level, or maybe only produce under certain conditions. So for example, if you use the promoter upstream of the ligase gene in the lag operon, which we have learned, by the way, I forgot which chapter, but we have definitely learned this, lag operon. If you use that ligase gene promoter, if you supply lactose to the bacteria later on, the bacteria will express the target gene. How cool is that? If you take away the lactose, it will stop producing the target gene. So in a sense, lactose is like a way to switch on and switch off the transcription, and you can control when the transcription occurs. So how cool is that? It's like a switch. So yeah, that is promoter. So origin of replication, restriction site, promoter. Last but not least, this is foreign to you, marker gene. Now before I talk about a marker gene, think about this. If you go to your kitchen and you see a bottle, maybe with like an unknown white substance, like of powder, you don't really know what it is until you taste it or try it. But what your mom do? Your mom would probably take a marker pen and a sticker and write salt and stick it on the container. Or maybe sugar, right? Label it sugar using a marker pen. Now, that is called marking, right? Marking your food. So, marker gene, same idea. A marker gene will tell us whether the recombinant plasmid is present in the modified bacteria. It also can help us identify transgenic organisms. Usually, the gene of interest is inserted close to or into the marker gene. You can see here that the marker gene in this diagram is very, very close to target gene. And in this case, the target gene and the marker gene are expressed together. When the marker gene is expressed, okay, this could be a few things. It could be antibiotic resistant, gene for fluorescence, or easily stained substances. This would allow the bacteria to exhibit certain characteristics. For example, if you use the genes for fluorescence, okay, if the bacteria has that particular plasmid, then the marker gene will be expressed, and therefore the gene, the bacteria will glow green color, maybe. This is the same idea for antibiotic resistance. So the marker gene could be antibiotic resistant. So if the bacteria has the particular modified plasmid or recombinant plasmid, then it would be resistant to antibiotics and grow even though the antibiotic is in the environment. Okay, we will talk about this in much more detail later on, but this is what it is just the idea you need to know. Marker gene is there to help us identify the recombinant plasmid later on. So yeah, we have answered the question now. Why plasmids? This is why. It sounds complicated, but it's actually okay. Just calm down, breathe, take deep breaths, and think of it as 4 items. Alright, so where were we? First, we obtained the mRNA of the insulin gene, then we converted it to DNA. Then, we cut it using restriction enzymes. And we cut the gene, both sides, as well as one side at the plasmid. As a result of that, we form identical sticky ends. Oh, sorry, complementary sticky ends. My bad. Complementary is a much better word. Don't use identical. Complementary sticky ends. And these sticky ends are also complementary here. Okay, these are all complementary. Okay, let's scrutinize it a little bit. You can see this one. It's AATT. This one can bind to TTAA on this side in the plasmid. Complementary base pair. Okay, let's change color. On this side, it's TTAA. This side we don't want already. This one don't want already. Just want the target gene, right? So TTAA, again, unpaired sticky end here, it's complementary to this side, AATT. And these complementary sticky ends, these two here, complementary sticky ends, can only happen if you use the same restriction enzyme. Same one. If you don't use the same one, the sticky ends produced will not be complementary. Okay, so if they're complementary, they can form hydrogen bonds with one another. But hydrogen bonds is spontaneous. But phosphodiester bond is not spontaneously formed. It needs an enzyme. And the enzyme we're going to use in the next step is DNA ligase. This is the pasting mechanism. To recombine the gene to plasmid DNA, you mix it together, use DNA ligase to seal in the nicks in the sugar phosphate backbone and catalyze the formation of the phosphodiester bond. Right? This occurs, okay, you can see that this target gene is in the plasmid now. This occurs at this point and this point. Hydrogen bonds can form again between those pairs, but here you need the phosphodiester bond right here. Same for here and here. Phosphodiester bond here and here. So use DNA ligase for that. And ta-da! Now you have recombinant DNA or recombinant plasmid. Which leads us to our fourth point. We need to now insert the recombinant plasmid into bacteria itself. So how do we do that? First of all, we want to mix the recombinant plasmids with the bacteria. Then we treat the bacteria with a solution of calcium ions and allow it to cool for a moment. And then there are two ways after that. We can use heat shock, which is not very hot, 42 degrees only, not very hot, for 30 seconds. Because we don't want to kill it, we just want to shock it. Or we can use electric shock, again, very small sort of electric shocks. You can see the details here, you don't need to remember it though. This process is called electroporation, and what happens is this allows the cell, the wall of the bacteria and the cell membrane of the bacteria to become temporarily unstable. So this increases the chances of plasmid passing through the cell surface membrane of the bacteria and into the bacterial cell. As I said just now, only 1% of the bacteria will take up the recombinant plasmid. It is rarely successful. It takes a lot of effort and time in order to make sure this works. Okay, it will only work for 1% of bacteria, and this bacteria that has taken up the recombinant plasmids are considered to be transformed, or we can call them transformed bacteria or modified bacteria. Because there's only 1% that has succeeded, we need to check. We need to identify which one is transformed and which one is not. Because after you mix them, you can't really tell. They're all in like one test tube or something. You can't tell. So how do you identify them? Marker genes that we talked about just now. It helps to recognize recombinant plasmids, modified bacteria, or transgenic organisms. So there are three types again: genes for antibiotic resistance, gene for fluorescence, or genes for easily stained substances. The gene of interest is usually inserted close to or into the marker gene, and usually both they are expressed, but not always, usually. So you can see here, gene of interest is inserted close to the marker gene in this area, so the target gene and the marker gene will both be expressed under the same promoter. So they'll both be expressed in the bacterial cell. But when the gene of interest is inserted into the marker gene, so it happens that the restriction site is here, into the marker gene, this disrupts this marker gene. So the marker gene switches off instead of switch on. Usually there's another marker gene at the site here, you can see, which is not disrupted. And you might think, okay, why is this unnecessarily complicated? And that's because it has an extra function, which might be very useful to us. The extra function is that it's easy to identify the recombinant plasmid this way. I guess you may be wondering, like, isn't the recombinant plasmid like for sure? Well, not really. There are a few ways things can go wrong. Okay, it's either number one, the bacteria did not take up the recombinant plasmid. So, like, the bacteria didn't take it up at all, so the bacteria doesn't have it lah. Or the bacteria did take it up, but the gene of interest did not join the plasmid itself. The plasmid just rejoined with itself. It then joined with the gene of interest when we pasted it back just now. There's no way to tell unless we use the marker gene, use the double marker gene. Okay, one disrupted and one not. So let's start with antibiotic selection as a way to identify transformed bacteria. But after that, we'll talk about using the green fluorescent protein, so how to make bacteria glow green color. And the third one would be how to use an easily stained substance like beta-galactosidase and Gus. Okay, one by one. Let's talk about antibiotic selection first. So antibiotic selection is one of the examples that uses two marker genes. Okay. For example, right, ampicillin resistance and tetracycline resistance. So here we have ampicillin here and tetracycline resistant gene here. Okay, and the target site for BAMH1 is here. So this is actually a restriction site. And you can see here the result of that is that the insulin gene is inserted into the tetracycline resistant gene. So tetracycline resistant gene is actually switched off because of this disruption, whereas the ampicillin resistance gene is kept intact. Okay, so what do we do in this in antibiotic selection? We know that this happens. This is a recombinant plasmid. Okay, so what do? The bacteria that we tried to transform just now, 1% success again. We take it, we grow it on an agar containing the antibiotic. Let's start with ampicillin first. Okay, so we want to see which one has the plasmid. So if it has the plasmid, it will be able to survive. So these are the 5 colonies here that are able to survive because it has the plasmid. But it could have this plasmid or it could have this plasmid. You don't know which of these plasmid it has, right? So we gotta make a replica plate. Usually we use a sponge or velvet pad. This is a velvet pad, I think tied on a cork with some cloth and tape maybe, or rubber band. Then just chop, basically like transfer the colonies by making an imprint and then stamping it on a new agar plate, this time with tetracycline. And since the tetracycline plate is it has tetracycline, and we know that the tetracycline resistant gene, it's switched off because it's disrupted by the human insulin gene, the bacteria with the recombinant plasmid will actually die. That's okay, we have spare. But the bacteria of the recombinant plasmid will actually die. So you know that, hey, these 2 colonies that did not survive, these are the ones that showed success. Okay, so let me sort of break it down for you. If the bacteria doesn't have the plasmid at all, so no recombinant plasmid, no plasmid at all, it will die in the ampicillin. It will also die in the tetracycline. If the bacteria has the plasmid, but it is not recombinant, that means something has failed when we stuck the gene didn't insert, it just didn't. So the plasmid just closed back on itself. So if it has a plasmid, it's not recombinant, it will survive ampicillin. Okay, that means it has this particular plasmid. This one, right? And it will survive tetracycline. However, if the bacteria has the plasmid and has the recombinant, especially the recombinant plasmid here, like the insulin gene here, this means it will survive in the ampicillin, but it will not survive because of this disruption. It will not survive in the tetracycline. So you can take these 2 colonies from ampicillin plate, which are probably still there, and you can go and grow it some more. Okay, so now that we have identified that, great, no problem, right? We can just go on with our lives. But you might be thinking, okay, antibiotic resistance, isn't that like bad? Like, why are we using it? And you're right, like, this antibiotic selection is incredibly useful because it helps us check whether the recombination has worked and the plasmid is there and it's great. But problem, risk of spread of antibiotic resistance. If you remember, back in AS, we learned that antibiotic resistance can spread to other bacteria of the same or different species via conjugation. Or you think of it as sexual reproduction. It's not, but it's conjugation. Okay. This makes the use of antibiotics less effective in disease-causing bacteria if disease-causing bacteria happens to obtain this antibiotic resistance gene. And this results in slower process for identification of transformed bacteria as well, because eventually the bacteria that is not transformed are also resistant in the first place. So that is a problem. So the only solution here is just to use other methods. Don't use antibiotic selection so much, although it's great. Don't use it so much. Let's find other alternatives. So they did. They found green fluorescent protein. They found a way to make things glow. This is actually incredibly beautiful. So, GFP, green fluorescent protein, GFP gene is actually an example of a gene of fluorescent substances used as a marker. It's from jellyfish, and GFP actually emits a green bright light when exposed to UV. So this is dark because it's under UV conditions, and usually it's in like an isolated room that is dark, that only in little UV box. There are safety mechanisms. If you go to uni and do bio, you will see it. Anyways, bacteria with the plasmid will express GFP and appear green. And therefore, you can take this bacteria that express GFP and you can go and make more of them later on. Okay, next. Well, that's a spoiler, but never mind. You can use easily stained substances. So there are two types here, two examples. Same thing, but two examples. These easily stained substances actually is not in the gene itself. The gene usually codes for an enzyme. So gene for beta-galactosidase, aka the ligase gene, very useful. Or the gene for the Gus enzyme is used. Now in these cases, right, the enzyme splits a special non-blue substrate into a product that is blue. So this special non-blue substrate, you don't need to know the name. You can feed the bacteria a bit, and then the ones with the plasmid will become blue. So you take all these blue spots, and then you go and make more of them. So yeah, that's how you identify the bacteria with the recombinant plasmids. There are advantages of using fluorescents and easily stained substances as markers. Not only that it avoids use of antibiotics in the first place, it's actually more economical, time-saving, and labor-saving because it glows in the dark. You don't need to like put antibiotics in, put antibiotics out, transfer. It's just glows in the dark. You just put it under UV light. It's just easier. It's a visible color, so you don't need to like see which ones survive, which ones not. It's just like very bright and very visible. And it enables identification of not just transformed cells and bacteria. If you take this gene, this plasmid, and you insert it into other animals can light up too. So we have pigs that glow here. This is literally called glow pigs. There are animals that glow here. Okay, you can use this to identify certain genes in other animals as well, using the marker gene. We'll not go into details. And there's no known risk or ill effect on the GM organism, which is good, because it can only be visible under UV light. So under normal light, it doesn't look like anything. Okay, so we cut, we paste, we identified the bacteria. Now we're gonna grow the bacteria. Since we found the bacteria that is modified, we can put it in a fermenter. we clone it, we multiply it, okay. Again, E. coli can divide once every 20 minutes, almost half an hour. And basically, bacteria will have the replication, transcription, and translation machinery to copy a gene and express the protein products. In this case, we're talking about human insulin. So it can produce multiple copies of the gene for human insulin as well as the human insulin itself. And this can be extracted and purified and sold on the market. This is great. This is so good, because this means that we do not need to use the conventional method anymore. The conventional method is, extracting insulin from pancreas of pigs or cattle, right? Not very halal, right? But that used to be the case, but now we can produce. We don't need to harm the pigs and cattle, for one. We can produce human insulin in bacteria, which is chemically identical to human insulin. Pigs and cattle may not have chemically identical insulin to us, right? So this means it has faster response, fewer side effects, less risk of allergic reaction, it does not stimulate the immune system, and it's an exact fit to insulin receptors. It's all great. And some people, though, have developed tolerance to the animal-derived insulin. So even if you are tolerant to the animal-derived insulin, this human insulin produced in bacteria will still be effective for you. Obviously, there's no ethical issues here, and there's no ethical issues related to religion as well. There's no killing of animals here. It's lower cost for purification and processing, relatively compared to pigs and cattle, and it's mass production. Pancreas of the pigs and cattle. How many pancreas do you need to get in order to isolate enough insulin? Using bacteria, we can have a large and constant reliable supply all year round. It's mass produced. And less risk of contamination and disease. From pigs and cattle, there might be diseases, that can transfer to humans, but in this case, don't have. And last but not least, not the main point, but there is a potential to engineer or improve on the recombinant proteins we have. If we don't like it, we can change a few bases of the gene and then make more of it using bacteria anytime we want. It's very versatile. So yeah, these are the advantages of producing human insulin by recombinant gene technology. Crazy, right? Amazing. Now, you might be thinking, what are the applications of this? Well, as you saw just now, it's great for production of pharmaceuticals, not just insulin, many things as well. However, there's no modifying of protein in bacteria because they don't have membrane-bound organelles. So far, all the recombinant DNA technology we have been using with bacteria has to be proteins that are not modified. However, we can genetically modify eukaryotic cells, okay, for example, yeast cells, insect larva cells, mammalian cells. Bacteria is there, but ignore it. Okay, and we can use these cells to produce human proteins as well. And you can use those cells in order to modify the protein, add carbohydrates to it or whatnot. So this is a list of human proteins that are produced in different things. So we have human insulin, which is treatment for diabetes, produced in recombinant bacteria. We already know that. Factor VIII, which helps blood clot in hemophilia patients, and this is produced in GM hamster, kidney, and ovary cells. GM means genetically modified. This is kidney and ovary cells in the fermenter itself. So yeah, it's a eukaryotic cell. We can be using adenosine deaminase, or ADA, to help in development of B and T cells in severe combined immunodeficiency disorder patients, SCID patients, or SCID, what they say, that's how they say it. They have really bad immune systems, like, I will tell you more about it later, but basically their immune system is like not functioning at all, and they have to stay in like a very sterile environment at all times. Like, you cannot go out, you just stay in quarantine forever. But there is treatment now for it, adenosine deaminase, which helps And this is produced in genetically modified insect larvae of the cabbage looper moth. And in this case, yeah, the cabbage looper moth actually like lays eggs and lets it develop to larva, and those larvae has this particular drug in it that helps people, so it's very important. Of course, there are other things like antithrombin that stops blood coagulation in milk of GM goats, and this is important in like stroke patients or people with high blood pressure. Okay, alpha antitrypsin for emphysema in milk of GM sheep. So we can produce goats and sheep that produce milk with drugs we want in them. How cool is that? Alright. But we'll talk about genetically modifying animals later. For now, let's focus on the recombinant DNA technology. You need to remember that it's production of pharmaceuticals. You need to remember these 3 examples highlighted in red and what treatment it is for. Last but not least, there's one more application, and it's quite big, so I'll do it in the next video, but just a teaser. Recombinant DNA technology is used a lot in genetic engineering to produce GMOs in agriculture. So, to find out more about that, I'll see you next video. Bye!